Hello and welcome back to Educator.com and welcome back to Biochemistry. So today we're going to start our discussion of enzymes. I can't even begin to overemphasize the importance of enzymes. I mean, enzymes are biochemistry. So a really, really good understanding of enzymes, how they work, some of the quantitative aspects, things like that, absolutely essential to everything that goes on in biology. So let's just jump right on in and spend a fair amount of time with enzymes. Okay. Now, having said that, enzymes, in the discussion of, see what's going to happen is we're going to introduce enzymes, talk a little bit about their general properties, a little bit about how they work. We're going to give some examples of some enzyme reactions, and we're going to talk a little bit about the quantitative aspects, some of the kinetic aspects, things like that. But this whole idea, we're not going to get too much into it. We don't want to say too much at this point. This is one of those topics where you do run the risk of saying too much that's unnecessary. We just want to give you a good, solid grounding in enzymes. And once we actually start to talk about the metabolic pathways, glycolysis, fatty acid breakdown, amino acid breakdown, citric acid cycle, things like that, we'll be revisiting the enzymes. So a lot of what we ultimately want to learn about enzymes will come from all of the exposure that we get when we deal with the enzymes in the individual pathways. So having said that, let's see what we can do. So enzymes are, again, they are everything. So enzymes are proteins, very, very large proteins. So enzymes are proteins. The only exception are the catalytic RNAs, which you'll end up discussing in a subsequent class, molecular biology. Catalytic RNAs. Okay. Now, some enzymes function as is. Some enzymes function as is. That is, without additional chemical groups to help them along. So without additional chemical groups to make them work. Others require something called a cofactor. Excuse me. Let's get comfortable here. So others require something called a cofactor. So a cofactor can be one of several things. It can be an inorganic ion, you know, maybe like iron 2+, or maybe magnesium 2+, or manganese 2+, etc., zinc, cobalt, something like that. It can also be an organic molecule, an organic or occasionally a metalloorganic molecule called we usually call it a coenzyme because it is an actual molecule. It's not just one single metal ion, called a coenzyme. Coenzyme. Now, some enzymes require both ions and coenzymes. Okay, now, when an ion or you know what, let's just call it a cofactor. Okay, all these names that we're throwing around. I know it can tend to be a little bit confusing. The names themselves are not altogether that important, except for the occasional quizzes that you take. What's important, again, is the chemistry, just understanding what the function of these things is. So when a cofactor, either ion or coenzyme, is tightly bound or covalently attached, to the enzyme, we usually call it a prosthetic group. Okay, now let's see. The entire, let's try this again. Enzyme plus cofactor is called a holoenzyme. Again, just a bunch of nomenclature that's not altogether that important. So when you have the enzyme plus its cofactor, the thing that's all ready to go, it's called a holoenzyme. The enzyme part alone, without its cofactor, it's called an apoenzyme. Enzyme without its cofactor is called an apoenzyme, or sometimes an apoprotein. Okay, now most coenzymes they derive from vitamins. They derive from vitamins. So some examples would be, well, let's actually take a look at some. So we have this one right here. This first one, this is riboflavin. Okay, this is one of the vitamins that we need to ingest in order for bodies to function properly. And the enzyme that is derived from riboflavin, this part right here, you notice it's right here. Okay, this looks like flavin adenine dinucleotide. That is the coenzyme form of the riboflavin. We ingest the riboflavin, the body converts it to FAD. This is the coenzyme. This is the active part that works in concert with the particular enzyme that it's attached to. Another one would be, another vitamin would be pyridoxine. And pyridoxine is the vitamin that we ingest. The coenzyme itself is something called PLP. It's called pyridoxal phosphate. Pyridoxal phosphate. And again, we'll be running into these. We'll be getting into more details later on when we speak about specific enzymes. We're going to talk about these cofactors, what they do, how they do it. We're going to get into, detailed mechanisms and things like that. Right now, just wanted you to see some examples. Let's see, a couple of other examples. We have carbonic anhydrase. So, just wanted you to see, actually in this case, a ribbon diagram of the actual enzyme itself. And I don't know if you can see it here, that little zinc ion. So that is a prosthetic group it's a cofactor for carbonic anhydrase, happens to just be a metallic ion. And this, of course, is a close-up view. This is zinc ion. You see it's coordinated to a histidine, to another histidine, to another histidine, and it's also coordinated to a hydroxide ion. So within the enzyme, within the folds of the enzyme itself, there is this, what you might call a partial active site. This particular zinc ion is coordinated to 3 histidine residues and a hydroxide. That's it. Nothing particularly strange going on here. Okay, so now I'll just write close up here. Close up of the carbonic anhydrase. Okay, now enzymes are classified according to the reactions they catalyze. That makes sense. To the reactions they catalyze. Excuse me. Okay, an international system has been agreed upon. System has been agreed upon. Agreed upon. But be aware that many, if not most, if not most common names are still in regular use. So most of the time we'll be referring to things with their common names. We'll be calling them, for example, alcohol dehydrogenase. We won't be calling it alcohol NAD oxido reductase. That's going to be the formal name. We'll just, for the most part, we're going to be using common names. Now, clearly, a lot of the enzymes that were discovered early on, before the, before the international agreement of systematization, those common names, I mean, they're just so entrenched in the literature, they're just so entrenched in our daily use that they're never going to go anywhere. So they have formal names, but it's the common names that you're going to, see them as. The more recently discovered enzymes, those are the ones where you're going to actually use the systematic name instead of the common name, because no common name exists for them. Okay, so common names are still in regular use. So let's see here. Let's go ahead and move on to this. So each enzyme, so each new enzyme or each enzyme is given a number. It's called the enzyme commission number. The enzyme commission number and a systematic name. So it's given both a number and a name. Now, the EC number has 4 digits. The first number specifies the class of the enzyme. Specifies the class, the enzyme class. There are 6 classes. So an example would be, and then of course you have your subclasses underneath that. Example would be, let's just take the most basic one, EC 1.1.1.1. So class 1, these are enzymes, and they are called oxido reductases. They are involved in electron transfer. Oxidation, reduction. They're involved in electron transfer, facilitating the transfer of electrons from one molecule to another molecule, from one substrate to another substrate. The transfer of electrons. Transfer of electrons. So that's it. That first number, that's what that specifies. So let's go ahead and look at a systematic name. So in this particular case, the EC 1111, its systematic name is going to be, as we said, alcohol NAD plus oxido reductase. Oxido reductase tells me there's a transfer of electrons. Alcohol, the electrons are transferred from the alcohol. There's a colon, they're transferred to NAD+. That's what this means. From here to here, oxido reductase. Well, the common name is alcohol dehydrogenase. Alcohol Dehydrogenase. This is the enzyme that's responsible for metabolizing the alcohol that you drink, so it doesn't poison you. Alcohol dehydrogenase. Okay, so now we have the 1, 1, 1, 1. This tells me its class, an oxido reductase. Okay, this one right here, it tells me that it's acting on an alcohol group, a COH group. This one right here, the third number, it tells me that the electron acceptor is either NAD plus or NADP plus. And this one right here, it's specifically NAD plus. If you had 1.1.1.2, that would be alcohol dehydrogenase NADP plus. That's it. So really ultimately what's important, are all of the numbers. You can look all of these up. It's not a problem. You don't have to know any of these. It's really important to know that the first one is what specifies the class. And your teacher will probably ask you to know at least, the 6 classes of, so you have oxido reductase, you have lyase, you have ligase, things like that. So that's all that's going on here. The particular reaction that this enzyme catalyzes is the following. See, it takes a primary alcohol. So the enzyme binds 2 things. It binds the alcohol and it binds the NAD+. It brings them in close proximity and it allows the transfer of electrons and hydrogen from the alcohol to the to the NAD+ to produce the following. What the enzyme ends up releasing, you end up turning the alcohol into an aldehyde, and then you've also created this thing. That's it. That's it. Basically all you've done is what this enzyme done is it rips away that hydrogen, it turns this single bonded oxygen into a carbonyl, and then it releases something called NADH and H+ into solution. Okay. So alcohol dehydrogenase. Now, of course, you probably already know this. If not, ACE is the standard ending. Standard ending for an enzyme. So, alcohol dehydrogenase, pyruvate dehydrogenase, something isomerase, hexokinase. ACE, that's the enzyme. In this particular case, the cofactor, this enzyme does have a cofactor. The cofactor happens to be that molecule. So, and in this particular case, it's a cofactor that is not tightly bound to the alcohol dehydrogenase. It's actually free to float around. So we don't call it a prosthetic group, but we do call it a coenzyme. Okay. Now, enzymes are catalysts. Okay, let's go to blue here. So enzymes are catalysts. That's it. They're catalysts. All they do is they speed up a reaction. They speed up a reaction, but they do not affect the equilibrium position of a reaction. That's very important. They allow a system to come to equilibrium faster. They just don't change the equilibrium position. So again, there are many reactions that can take place. For example, if I want to, if I put oxygen and hydrogen in a container, well, thermodynamically, that's a very, very, that reaction wants to happen. It's very thermodynamically favorable. The problem is kinetically, it's not, because it has a very high activation energy. It takes a lot for it to go over that initial hump in order to become water. So thermodynamics and kinetics are not the same thing. A catalyst does not affect the equilibrium. It affects how fast it reaches equilibrium. That's it. That's all a catalyst does. So So it just speeds things up. Now, under physiological conditions, under physio conditions, bioreactions, they tend not to happen. In other words, they're not too thermodynamically favorable. They tend not to happen, or they happen very slowly. Again, this is why enzymes evolved. They evolved to allow for life processes to function without having to wait forever. It's really kind of amazing. Many of the reactions that give rise to life shouldn't happen, wouldn't happen. Enzymes allow them to happen. Absolutely extraordinary, powerful, powerful things. It's a lot more than just speeding up a reaction. It's the extent to which they actually allow reactions that are so complex to actually take place. Reactions that would never happen in the body, but enzymes allow them to happen. So, okay. Now, it does this. It does this, in other words, allow the reactions to happen by providing a place, an actual physical place, and a context, an environment for the reaction to take place. We call this place the active site. Very, very important. So where a particular something happens in the enzyme is called the active site. It's where the reaction takes place. Now I want you to think about this. You've got a whole bunch of, you've got just billions and just millions, thousands, whatever, different types of molecules floating around in your body in any given space. In order for a reaction to take place, well, a couple of things need to happen. The most fundamental of which is that, let's say if you have to have this molecule react with this molecule, well, it's like being in a hugely crowded city. How is it that one molecule on one side of the city is supposed to run into one molecule in the other side of the city? How the hell is that going to happen? They're not going to happen randomly. Or even if they do happen to meet randomly, maybe the conditions are not exactly right for the reaction to take place. What an enzyme does is it brings together this person or this molecule, that molecule. It puts them in the same vicinity. It holds them close together, and it provides a context for them to actually go ahead and have a reaction. That's what enzymes do. Bring together molecules, provide a space for them to actually do what they're supposed to do, which is react. In other words, align them properly, change this, change that, and then facilitates that reaction by making it a little bit easier for the reaction to happen, by possibly providing it an alternate pathway, a pathway that it wouldn't have otherwise thought of if they just happened to run into each other randomly. That's the whole idea behind an enzyme. It provides a place and a context for this to happen. We call that the active site. Okay. Now, the molecule or molecules an enzyme binds is called a substrate. It's called the substrate. You might have more than one substrate. It just depends on your particular point of view. If you want to consider, like in the previous example with the alcohol dehydrogenase, well, the primary alcohol is one of your substrates. You can consider the NAD+, you can call it a cofactor if you want. Well, it's free to float around, so it's also a substrate. It's the second substrate. Again, a lot of these things happen more to do with names. Names are not altogether important. What's important is what's happening. So in this case, it brings together the alcohol and the NAD+. In order for them to do what they need to do, then it sends them off on their way. The alcohol leaves as aldehyde, the NAD+ leaves as NADH, and the enzyme goes and does it for another pair of molecules. Okay, so the molecule or molecules an enzyme binds is called the substrate. Let's see. Okay, now the active site You guys already have a sense, I think, of what enzymes are. So the active site is often a pocket on or within the enzyme. Within the enzyme. Okay, and because an enzyme is a protein, this pocket is lined with amino acids which bind the substrate, with weak interactions. And so basically you have this pocket, and along the pocket there are these amino acids which are part of the protein, part of the enzyme. They interact with the substrate in certain ways with weak interactions, hydrogen bonding, ion dipole, hydrophobic interactions, and they sort of form a little bit of a cocoon, a little support for the particular substrate, and then it allows it to do whatever it does. We'll be talking a little bit more specifically about binding and weak interactions and things like that. But, okay, so amino acids which bind the substrate with weak interactions. Okay, often the substrate is completely sequestered from solution, is completely sequestered from the environment, from the aqueous environment. So in other words, when the enzyme binds the substrate, it doesn't allow water or anything else to come in. It provides a space for only that molecule and whatever else needs to be there for the reaction to take place. So that's what we call sequestering. It separates it from solution, so you don't have a bunch of other stuff floating around. So let's go ahead and just take a look at some of these things just to get a sense of what they might look like. So let's see, here we have an image of 3-dihydroquinate synthase. In this particular case, we have three substrates. We have three things that are attached. Some of the substrates are cofactors, some of them not. Again, just things that are attached to it. We have here, if you can see it, this thing is zinc. It's a zinc ion. And over here we have another substrate that looks like it's going to be the NAD+. And over here is the other substrate, the actual substrate itself, something called carbophosphonate. And again, you notice on here, this is a sort of a surface image of the enzyme. You notice that this particular binding pocket happens to be on the surface. This one, the substrate, seems to be actually inside, so it's a pocket inside of it. The enzyme has sort of closed itself around it, so it's not necessarily on the surface. And it looks like the zinc is on the inside too. Here, the second image it shows a close-up of a substrate, which is this thing right here, and some of its interactions with the amino acid residues that make up the active site. So here you can see that here's oxygen on here. I don't know which molecule this is, but it looks like it's interacting a little bit with this thing right here. You know, looks like maybe a carboxyl group, maybe a glutamate, something like that. And notice this is attached to the protein. So this is the protein, the amino acid side chain. It's the amino acid side chain that's actually interacting with weak interactions with the particular substrate. And that's it. And another sort of, another view of it. Here we had sort of a surface image. Here we have a ribbon diagram. Again, you can see the substrate is actually right here. And it looks like it's actually deep inside of this particular enzyme. It looks like maybe the enzyme opened up, substrate came in, and it closed itself around it. That often happens. We'll be talking about that a little bit later too, something called induced fit. That's it. That's all it is. Active site, enzyme, sometimes on the surface, sometimes inside the enzyme. Nothing particularly strange going on. Okay. So let's talk a little bit about how catalysts do what they do. So catalysts work by lowering activation energies. That's it. You know from general chemistry that that's what catalysts do. Activation energies and or providing an alternate pathway. I don't know. The thing is, by providing an alternate pathway, you're lowering the activation energy. So shall we say lowering the activation energy and or providing an alternate pathway? Let's just say catalysts work by lowering activation energies. How's that? And let's just say and providing alternate pathways. Alternate pathways for the reaction to proceed. Okay, so in terms of an energy diagram, let's take a look here. So we might have that, and we might have that. Something like this. So this is our substrate or reactant. Let's just go ahead and call it a substrate from now on because we want to get used to this nomenclature. And this is going to be the product. So this difference right here, the difference between here and here, this is something called the activation energy. This is an energy hump that the substrate has to get over before it's in a position to actually fall forward and move on to product. And this right here, this is called the transition state. So it has to reach, well, you know what, let me do this in red. So this right here has to reach the transition state. And often this energy barrier, this activation energy is very, very high. This energy barrier is what controls the rate of the reaction. The lower that energy barrier, the faster the reaction is going to go because more molecules are going to have enough energy to get over that hump. So this right here, this path, this is the uncatalyzed reaction. Okay, this difference right here, the difference in energy from here to here, this is the normal delta G. That's the thermodynamics of that. Now, the catalyzed reaction might go something like this. So notice, this and this haven't changed. We're still here and here. The delta G hasn't changed. Thermodynamics doesn't change. What has changed is this. Now, your activation energy is a lot lower. Because the energy is a lot lower, the reaction can proceed faster. That's what a catalyst does. That's all a catalyst does. It is not used up in the reaction. A catalyst itself, it goes in one form, it comes out of the reaction in the same form. In between, it might experience some changes, but itself is never changed from beginning to end of the reaction. All it does is it provides a different pathway for the reaction to proceed. That's all. That's how a catalyst does what it does, by reducing the activation energy, the energy it takes to get over the transition state. Okay, now let's go ahead and see. Well, let's do one more energy diagram here. Okay, this is substrate, this is product. Let's go ahead and make that here. Actually, you know what? Let me go ahead and, since I used blue for the uncatalyzed, I'll go ahead and keep it as blue for the uncatalyzed. Here, like that, and we might have something like this. Something like this. So here what we have is we have enzyme plus substrate. It's going to form something called the enzyme-substrate complex. Once the substrate binds to the enzyme, now we have this thing. Actually, you know what? Let me think I'm going to do 2 hills because I need 2 hills. 1, 2. And then we'll go there. How's that? So here, this is our enzyme substrate. Enzyme substrate. And here we have our enzyme product. Enzyme product. And of course, the enzyme is released and the product is released. So the enzyme and substrate come together. They form something called the enzyme substrate complex. The substrate is converted, changed to product. Now we have the enzyme product complex, and then the enzyme and product separate. The product is released, and enzyme goes back to start a new cycle. It can be represented like this. The substrate starts here. This first hump, in order to get to this first intermediate, this intermediate is the enzyme substrate complex. And then it goes to the next hump, enzyme product complex, and then the next hump in order to actually release this. So here, well, I'll tell you what, let's go ahead and just talk about it. So intermediates are short-lived viable species in a reaction pathway. Now, in this case, by pathway, I mean individual reaction in a, you know what, I won't say pathway because we're going to actually use pathway in a different context. Intermediates are short-lived viable species, I'll say, in the progress of a reaction. In the progress of a reaction. So in other words, it isn't just, reactions don't necessarily happen in a single step. They happen in multiple steps for a given reaction. Each one of those steps has a little transition state that you have to get over, and each one of those valleys constitutes an intermediate. So an intermediate is something that is there. It is short-lived. It doesn't live very long. So in the case of an enzyme to an enzyme substrate, it has to go over a small hill to become an enzyme substrate. Now that's an intermediate. Now the enzyme substrate has to go over another little bump to become the enzyme product, and then the next bump in order to actually become the product. So again, we have to differentiate between intermediate and transition state. They're not the same thing. A transition state is a very small, fleeting thing that the particular substrate has to pass through in order to become the next thing. So an intermediate is a place along the way from substrate to final product, sort of you call it an intermediate product, if you will. It's something that it has to pass through. It's a low energy point, not a high energy point. Okay, now conversion from one intermediate to the next intermediate constitutes a reaction step. That constitutes a reaction step. The slowest of those steps is called the rate-limiting steps. So if a reaction takes place in 5 steps, and one of those reactions is actually the slowest, that slow reaction is going to control the overall rate of the reaction, because you can only go as fast as your slowest step. That's it. A chain is only as strong as its weakest link. The slowest of those steps is called rate-limiting. Makes perfect sense. Nothing strange here. Completely intuitive. Now, here we're talking about individual reactions that happen in multiple steps. Most of what we discuss when we start discussing the metabolic pathways in the second half of this course, we're not going to be discussing individual reactions with multiple steps. What we're going to be discussing are reaction pathways. So you have from starting substrate to final product, there are going to be different reactions. So it isn't one reaction with multiple steps. You're going to end up having a reaction, another reaction, another reaction, another reaction. We're going to be talking about reaction pathways, metabolic pathways. Of those metabolic pathways, you might have one of those reactions that is going to be rate limiting. So you can have reaction 1, reaction 2, reaction 3, and so on. Each one of those is going to be catalyzed by an enzyme, enzyme, and enzyme. So from here to here is one reaction. It may or may not involve more than one step in this one reaction, but the whole thing taken together is called a pathway. So let's definitely make sure we distinguish between the individual steps of a reaction, which may have one step or more than one step, and a pathway which involves multiple reactions. So we definitely want to distinguish from that. Most of the time we're going to be concerned with pathways. We're not going to be concerning ourselves with the individual steps of each reaction. Sometimes we will when we want to get into the mechanistic details of that particular enzyme. More often than not, we will not. Okay. So I will go ahead and stop this particular lesson here. In the next lesson, we will continue on, get a little bit deeper into discussing enzymes, and we'll take it from there and move forward. Thank you so much for joining us here at Educator.com. We'll see you next time. Bye-bye.